Dr. Yodi, we may begin. Thank you.
Thank you. Hello, everyone. Welcome to the second session of the Southeast Asia Regional Meeting on COVID-19 treatment and vaccine development. My name is Yodi Mahendra Datta from Universitas Gadjah Mada, Indonesia, and I have the honor to chair this session. Now, let me start with a figure, a number, which has caught so many attentions this past couple of days. That figure is 95. 95%. Final result from Pfizer's COVID-19 vaccine trial showed that its vaccine had a 95% success rate. Before that, we heard from Moderna, which announced that their vaccine offers 94.5% protection. And we can expect to hear more announcements as we know there are more than 200 candidate vaccines in the pipeline. And these are not just developed by companies, but we are also seeing scientists and academic health centers around the world working on potential treatments and vaccine for COVID-19 disease. So in this session, we will hear from AAHCI members in Singapore, Indonesia, China, and Australia about current situation in their countries and what efforts they are involved in national and global level regards to COVID-19 vaccine and treatment, as well as perhaps the expected timeline for distribution in their countries. Now, each of the panelists will present for about 5 to 7 minutes, after which we will have a facilitated Q&A session. You can type your questions in the Q&A feature at the bottom of the Zoom toolbar on your screen, and we will go through and deliver your questions to the panelists at the Q&A sessions. So to start with, let me take you to Singapore to meet our first panelist, Professor Eong Oi. He's a professor in program in emerging infectious disease, Duke National University of Singapore Medical School. He's also co-director of the Viral Research and Experimental Medicine Center, SingHealth, Duke NUS Academic Medical Center. He's been also working in the field of dengue for 20 years, and his research interest spans dengue epidemiology to molecular pathogenesis of arboviral diseases. He's a three-time recipient of the Clinician Scientist Award by the National Medical Research Council of Singapore. Professor Ong Oi, the time is yours.
Thank you very much, Professor Yodi. I appreciate the invitation to speak at this webinar. And I will make it quick since the time is very limited, of course. It's a pleasure to be able to be on this call with everyone. And I'm just going to give you a very quick overview of the COVID-19 vaccine that we're working on in Singapore. This thing's not moving. Okay. So this slide probably needs no introduction. We all know that there's a lot of candidate vaccines that are currently under development, and several of which are, finishing phase 3. And excitingly, as we have heard, that the early data from Moderna and Pfizer are all very encouraging. And different companies, institutes have used very different approaches, ranging from inactivated whole virus vaccines all the way to nucleic acids and protein-based vaccines. And amongst the nucleic acid, you've got both DNA and RNA, and it's the RNA vaccine that is it's exciting, both in terms of the fact that it's very new, that this thing is in the very early stages of development, and yet the two front runners, Pfizer and Moderna, have basically used this method to develop their vaccine with pretty remarkable results. And this is the headline taken off today's newspapers in Singapore, that it could be authorized for use in the US within weeks. So we're also working on an mRNA vaccine in Singapore, and in collaboration with a company called Arcturus Therapeutics. This is a biotech company based in San Diego, California, and they're primary focus has always been to use RNA as a way of delivering drugs, but we're now working with them to turn that into a vaccine. And this is a conventional construct, schematic representation of an mRNA vaccine. So you've got the gene of interest, and most of us use the spike gene, and then it's capped at a 5' end and it has a poly A tail. So a classical mRNA structure. So the one that we're working with Arcturus Therapeutics is self-transcribing and replicating. In other words, it behaves a little bit like a virus in that the RNA will replicate. And the reason for that replication is because it contains also a second gene, and the gene is that of an enzyme that replicates the RNA, and this comes from an alpha virus called the Venezuelan equine encephalitis virus. So this gene will code for an enzyme that will make the minus strand as a template to make more positive strands. So this will amplify in terms of the numbers once it gets into the cell. Besides just amplifying the full-length genomic RNA, the replicase will also make a subgenomic RNA. In other words, it will insert right in the middle, and it will just replicate this part of the mRNA to make the subgenomic RNA, which then allows the amplification of the spike protein to be expressed inside immune cells of our body, and that then triggers the immune response. Right? So that's the concept, that it's not just putting in mRNA, but it's also putting in an mRNA that replicates. And the idea there is that then there's some dose sparing effect, but we also think that it will lead to better immune response, but at least theoretically. And the reason why we chose to go down that path and why we wanted to make use of a replicating vaccine is at least to mimic some parts of arguably the best vaccine that we have in the world, and that's the yellow fever vaccine. And we've been studying that for some years now in Singapore, and trying to understand why this vaccine is so good, where one dose will give you 10 years, if not more, protection against yellow fever. And so just this slide just sums up some of the properties. We've worked out, why some people get side effects, what are the genes that are needed to make a good immune response, and what genes when turned on too early give rise to these side effects, and then what, from the viral replication point of view, what then correlates eventually with immunity, right? I'm not going to go through the detail in interest of time, but essentially these are all published works, and it was based on this set of information that we then worked with Arcturus to try and develop this COVID-19 vaccine. So this vaccine is currently in phase 1/2 clinical trial, and part of the reason why we were able to take this forward into the clinical trials partly because of the preclinical data. And this is just to sum up to show you that there's a potential that we could get this to work as a single dose vaccine, where if you give the mRNA vaccine, in this case it's called the Lunar CoV-19, and then a month later you challenge these mice, this is the human ACE2 transgenic mouse, with a SARS-CoV-2 virus. So post-vaccination, you see that there's a dose-dependent increase in the neutralizing antibody response in these animals. And then when you challenge with the wild-type virus, in the placebo control, you see the weight loss, you see the gain in the symptom score. In other words, the mice get sick and they die by about day 7. But in both doses of the vaccinated animals, they had 100% survival without developing any symptoms. And at the pathological level, there was also no histopathological changes consistent with infection. So where we are with this is partly because of the lessons learned with yellow fever and how we could evaluate those vaccine response at the genetic level, or rather at the genomic level. We were able to then pool all this data together and move from a preclinical phase to phase 1/2 trial in about 4.5 months. Important to note that this vaccine construct, a self-replicating mRNA vaccine, had never been in man before. Unlike the mRNA vaccine that Pfizer and Moderna had done, those have been in man before, and so there was no safety data. And so the preclinical development was then critical to convince the regulators that this could be safe in Singapore. So this is a snapshot taken from the London School of Hygiene and Tropical Medicine's website, where they listed all the vaccines that they're under development, and where we are today is over here. We hope to be able to finish phase 1/2 trial and get onto phase 2/3 trial in about a month or two. So that's where we're at. Thank you for the opportunity to present this work on behalf of all these people who really made the data possible. Thanks very much.
Thank you, Professor Eng. So just to remind everyone, you can start typing your questions in the Q&A box in the bottom of the Zoom. Now, so from Singapore, we fly to Jakarta, Indonesia, to meet Dr. Budiman Bella. He's a medical specialist in clinical microbiology at the Faculty of Medicine, Universitas Indonesia. His research is focusing on vaccine, including DNA vaccine, subunit recombinant vaccine, and viral-like particle vaccines. He is now member of the Vaccine Expert Panel and member of the Laboratory and Research Expert Panel National Task Force for the Acceleration of COVID-19 Response in Indonesia. Dr. Budiman, the time is yours.
Thank you, Prof. Jody. Hi, everyone. I hope that you can see my slide. Okay, so the topic of my presentation is about the vaccine that we developed at our university, especially at the Faculty of Medicine, Universitas Indonesia. Now, this vaccine is just one of the many vaccine vaccine platforms that is being developed in Indonesia through by research institutes and also universities. As we know that the SARS-CoV infection as a virus will stimulate immune responses that are not just in the form of antibody or in particular neutralizing antibody, but it also will stimulate cytotoxic T cell response and also helper T cell response that are also important for elimination of viral infection. Now we know that vaccination is very important. It increases immunity and provide protection. So at the university, at Universitas Indonesia, that's what we call our university, we developed 3 platforms: DNA, RNA, and also viral-like particle vaccines for SARS-CoV-2 infection. Now, with the DNA vaccine, so they all actually starts with plasmids. So we clone spike and also membrane genes that encodes for these proteins. And then for the DNA vaccine, basically you inject the vaccine directly, this naked DNA, and then it will produce antigens. In terms of the platforms that we develop, we want the DNA vaccine not only to express the spike antigen, but also will express viral-like particle structure. And with the mRNA vaccine, you know that this is a very popular vaccine. Of course, we try to get away from the patent, so we try to develop something else. Hopefully it will work. And the last one is the VLP vaccine that we will produce in CHO cells. Now, DNA, I will just focus my presentation today on the DNA vaccine. It is an attractive solution for the threat of emerging and re-emerging pathogens. And we can say it is a new technology for rapid development of safe and effective vaccines. There are advantages and disadvantages of this platform, of course. Now, the advantages are cost-effective and easy to produce, stable even at ambient temperatures, no pre-existing immunity, so can readily be used in multi-dose regimens, elicits both cellular and humoral immunity, and people say it is quite safe in human. Of course, there are still arguments about this. The disadvantages is quite major, actually. It has an inefficient delivery into cells and limited studies showing efficacy in humans. However, at our university, we think and we believe we still have to develop this vaccine. As we all know, mRNA vaccine, although it is very effective, however, we still have problems with the stability at ambient temperature. A big problem with Indonesia, an archipelago country. Now, with DNA vaccine, also it can be produced quite quickly. So the release and delivery is quite rapid in comparison with other vaccine platform. In this picture here, it is compared with egg-based production for influenza vaccine. As I have mentioned before, there is still a major drawback of DNA vaccine in terms of its inefficient stimulation of immune responses. And this is due to, we believe, delivery of DNA vaccine into nuclear compartment of non-dividing cells as one of the major causes. Now, DNA vaccine can only be expressed if it can enter the cellular nuclei. And in order for the vaccine to enter the nuclear compartment of a cell, it has to pass through the cytoplasmic membrane to reach the cytoplasmic compartment. And after that, it has to pass through the second barrier, which is the nuclear membrane, to reach the nuclear compartment. This picture here just shows that with mRNA vaccine, you can see that it only has to pass through the cytoplasmic membrane barrier, and then it can access the ribosome so that it can be, so that the mRNA can be used for translation of protein. In terms of DNA, in terms of vaccine, of course, we can translate the vaccine antigen here in the ribosome. Now, with plasmid DNA, it has these two barriers, the cytoplasmic membrane. After it has passed that barrier, and then it has to go into the nuclear compartment, having this barrier of intact nuclear membrane, especially in non-dividing cells. And after that, it will be transcribed into RNA, and then the RNA will be used for translation of the antigen. There are various delivery methods for DNA, but I just want to focus on the last one here, cell-penetrating peptide. Now, what have we done at Faculty of Medicine, Universitas Indonesia, to solve the inefficient delivery of DNA vaccine? We have developed a peptide we call as ALMR. It is a cell-penetrating peptide for DNA delivery into nuclear compartment of non-dividing cells that is more efficient than liposome formulation. Now, you can see in this picture here, this is the result of study conducted by one of our PhD candidate by that time. She's now a doctor, a PhD, Dr. Sylvia Trividianing Tias. You can see here that using the ALMR peptide, we can observe a very significant transformation of PBMC. Actually, we can say these are monocytes because these cells have been isolated through plastic adherence. Now, in comparison with just lipofectamine, it is significantly higher than using lipofectamine for transfection. So we have confidence that we may be able to use this for our DNA vaccination. And we have cloned the genes, of course, and confirm it by sequencing. And then we express it in mammalian cell culture and analyze it with immunostaining. Now, this is just a preliminary result of antibody stimulation in mice model. We can see here that using a liposome, this is the DMRI, liposome, 25 microgram DNA plus ALMR, it has significant increase in comparison with no ALMR. Even when we reduce the dose to 5 microgram, the ALMR still show significant stimulation of the antibody response. So I think that is just what I can present today. This is just a preliminary result, but we are hoping that we can increase the efficiency or effectiveness of this vaccine by introducing also self-replication ability of the mRNA produced by the DNA. Thank you, and these are our team. And I would like to say terima kasih for your attention. Thank you.
Thank you very much, Dr. Budiman. I would like to invite the attendees again to start writing down your questions in the Q&A box. Now we'll continue our journey to Shanghai to meet Dr. Yingji. She obtained her doctoral degree in experimental medicine at McGill University. She then became a postdoctoral research fellow in the vascular biology program at Boston Children's Hospital and Harvard Medical School in 2011. And in 2017, she returned to China and she joined Shanghai Jiaotong University School of Medicine and started her own lab in Department of Biochemistry and Molecular Cell Biology. Her research is mainly focused on cancer target discovery and mRNA-based therapeutics. Dr. Yingji, time is yours.
Thank you, Dr. Yudi, for your introduction. So now I'm sharing my screen. Okay, so it's my pleasure to present my work here. So we made a COVID-19 mRNA vaccine encoding virus-like particle. Because of a high mortality and rapid spreading associated with COVID-19, attention has been focused on the need to develop effective therapeutics and vaccine against SARS-CoV-2. And according to the WHO, now there is more than 150 COVID-19 vaccines are presently in development and with around 12 undergoing late-stage testing. So very recently, Moderna and BioNTech announced their mRNA vaccine candidate provide strong protection based on initial data from its phase 3 clinical trial. As shown in the table here, and besides these two mRNA vaccines, there are other 3 kinds of vaccine, including adenovirus, protein-based, and inactivated virus. So our lab carried out 2 studies in fighting SARS-CoV-2. One is in collaboration with Dr. Lin at Fudan University to develop mRNA-based vaccine. Another study is in collaboration with Dr. Ches at Hebrew University in Israel to screen FDA-approved drug library to find drug candidates that can block or inhibiting the viral entry. So today I will just present briefly our ongoing study of development of mRNA vaccine against COVID-19. So the core principle of mRNA-based therapeutics for vaccination and other clinical application is that to deliver the transcript of infect, as shown here, use a delivery platform such as lipid nanoparticle. So this mRNA encoding one protein or one more protein, such as pathogens, into the host cell, then use the host cell's translational machinery ribosome to make the protein by its own. And this translated protein can be within the membrane or secreted outside of the cell or located intracellularly. So mRNA is synthesized by in vitro transcription. And here is a structure of the mRNA. And we can see that there is a 5' cap structure and 3', and there is a poly A tail. The coding region is flanked by 5' UTR and 3' UTR. Very importantly, the chemical modification can significantly increase the stability and translational efficiency of synthetic messenger RNA, as well as can decrease the immunogenicity. As showing this video here, here is the two different kind of modification to show the encoding the same EGFP sequence mRNA. And the left one is with 5-methylated CTP and a pseudo UTP, and the right one is a 5-methyluridine. And you can see that there is a big difference of the protein expression dynamics and expression level. So like other vaccines, the mRNA vaccine worked by training the immune system to recognize a threat like virus and began to produce antibody to protect itself. In case of COVID-19, the mRNA encapsulated by the lipid nanoparticle and then inject into the cells, and the cells will produce the spike protein and on its own, the spike protein does not harmful, and it also triggers the immune response, including secretion of neutralizing antibody and the induction of T cell response. So in our study, we designed 3 type of mRNA vaccine candidate for COVID-19, which including the receptor binding domain RBD, the 4 lengths of spike, and the 3 structure protein SME to form as a cocktail to form a virus-like particle. To enhance the protein expression, and we did in-depth codon optimization of the protein, as well as chemical modification nucleotide screening. And we found that the pseudorin UTP gave the consistently highest level of protein expression. When we transfected this 3 mRNA SME into the cells, we can easily detect the protein expression by a Western blot. And when we collect the supernatant from the cells and visualize to use an electron microscope, we'll be able to see the virus-like particles. And those particles, as you can see here, the diameter is an average of about 100 nanometer with spike protein density decorated in the on the surface, suggesting that the SARS-CoV-2 virus-like particle have formed. We then use a well-established lipid nanoparticle formulation to package the mRNA and then inject the mice intramuscularly at the day 0 and did a boost at the day 21. And then we did an ELISA measurement to measure the spike protein specific antibody level, and we find that at week 3 we'll already see there's increasing of the antibody titer, and it's after the boost the antibody level goes up very dramatically and keeps quite stable till the week 8. And then we use a virus VSV system and use a pseudoviral EGFP reporter system to measure the neutralizing antibody titer. And as shown in the figure here, we can see that the spike protein group gives a neutralizing antibody titer, which is similar to the level that's reported by Moderna of their mRNA candidate. And importantly, our VLP group gave the neutralization antibodies level is about 2.5 fold higher than the S group. And at week 8, the neutralizing antibodies level is still increasing. As we know, the T cell response is also critical for vaccine-induced cellular and humoral protection against the future infection. So we also measure the T cell response. As showing the figure here, both spike and VLP group increase the frequency of the CD8 positive population, as well as enhance the CD8 positive cells activation. And when we measure the interferon gamma production, and we confirm that the VLP group gave induced very strong T cell response. So in summary, our study demonstrated that COVID-19 mRNA vaccine encoding virus-like particle can induce strong antiviral like immune response. And now we are going to do the virus challenge experiment in the non-human primate, and we are looking forward to see its effect. So our work is in collaboration with Dr. Lin at Fudan University and Dr. Xia at Xiamen University, and also incorporate with bio company RNAQ. And this work is also supported by the special funding from Shanghai Jiao Tong University. And now this is the end of my presentation. Thank you very much.
Thank you. Thank you very much, Dr. Yingji. Okay, so last but not least, we go to Sydney to meet Dr. Walczak. He's program manager of the National System Level Initiatives at the Mari Dulu Budiari Gumal, the Sydney Partnership for Health, Education, Research and Enterprise, or in short, SPHERE. So he supports SPHERE's engagement with the New South Wales State Government and the Australian Health Research Alliance of Research Translation Centres. He coordinates the SPHERE data-driven healthcare platform, and he works with SPHERE's partner organizations and academic and clinical leaders to progress implementation-focused clinical and health system research. Dr. Walczak, the time is yours.
Thank you. Let's quickly share my screen. I hope that's showing up. Okay, so thank you so much for the opportunity to speak about our work undertaken at SPHERE. So I'll begin by giving a brief overview of what SPHERE actually is. We're an Australian government accredited advanced health research translation centre established in 2015 and certified in 2017, and one of 7 similar centres around the country. Hence, I think my presentation will be slightly different to my colleagues today. We have more than 2,800 members across 14 partner organisations with over 180 external relationships with industry and academia and clinical care. We currently have over 40 clinical trials either completed or ongoing within the last 5 years since our establishment. And our activity spans the full translational pathway from basic science through to public health work. So throughout all of the work that we do, we focus really heavily on implementation and reducing the lag from discovery to practice. Our partner centres include 14 quite substantial research assets around the state, including universities, clinical service delivery networks and medical research institutes, each of which has its own track record of significant advancement in clinical care. But we attempt within our centre to do things slightly differently, moving away from the culture of siloism to bring all of our partners together to advance, advance research, development, innovation and implementation faster for the benefit of clinical care. Our work's organised into 16 clinical academic groups, spanning a variety of health conditions and populations across the life course. Each is thoroughly multidisciplinary and operates with a fair degree of autonomy. And this is backed up by 9 strategic platforms focusing on critical areas that are known to be necessary to support rapid implementation of advancements in care. So I'll begin by giving a brief overview of the treatment and vaccine context in Australia more broadly. So in Australia to date, we've had a caseload of 27,000 odd cases nationally, the majority of which have been in our largest state, Victoria. And this is against a population of over 26 million. So in the broad scheme of things, it's been a fairly low incidence here, which has proved both, quite challenging in terms of progressing a significant body of clinical research to address COVID. We currently have 6 registered vaccine trials nationally, primarily in the phase 1, some progressing into phase 2, the most advanced of which I believe is led from Queensland University, which is a protein-based vaccine. And we also have a variety of phase 1 to 3 therapeutic and intervention trials currently registered and recruiting around the country. Interestingly enough, there are no later stage trials of vaccines or major advances in treatment currently registered in Australia. Of those that are progressing, however, we have a number of multi-therapeutic platform trials targeting a variety of treatments. The critical takeaway, though, is that there's no emergent consensus in the work that's being done here as to what might constitute a best treatment, given the heterogeneity of patient response, symptoms, impact on organ systems, and the emerging body of literature on risk factors for poor outcomes. Our federal government, pleasingly, has been quite proactive in their approach to approving and bringing treatments to practice. They've stood up a number of expedited exemption, assessment and recall pathways to assist with getting new therapies into practice. And they've been quite proactive in securing vaccine dosage from 4 of the leading candidates around the world at the moment, subject to successful completion and regulatory approvals, totaling around 130 million doses across the country. Interestingly, they've progressed beyond securing those doses to also thinking about the distribution pathway for those once they become available, hopefully early in the new year, setting out a vaccination policy nationally just last week, focusing on priority distribution groups, ensuring that the vaccine is free to citizens, residents and most visa holders, and that there's sufficient data captured for surveillance purposes down the line, and clear delineation of roles between our federal government and state governments. In New South Wales, where Sphere is located, we have the largest population in the country. You'd have had a pretty small caseload, just over 4,300 cases. We suffered a pretty significant first wave outbreak, which has been controlled quite effectively through substantial public health measures such as contact tracing, critical communications with the broader public, hotel quarantine and wastewater testing. Our health system has been quite effective at managing the response needed in terms of increasing critical care bed capacity and transitioning many services to telehealth. And our low case count, as I said before, has made clinical research in our environment pretty challenging. That doesn't mean that we've been, completely absent from this effort. At the outset, we were expecting a caseload similar to what was being seen in Europe at the time, with 20 to 60% of the population projected to be impacted. Hence, we worked very rapidly with many of our partners across the research and the health and medical research sector and the state government to begin preparing a robust response. We began by compiling a statewide inventory of assets to assist with what was anticipated to be a heavy program of clinical work, and have engaged continuously with key networks within the New South Wales government, a vaccine advisory group, therapeutics and research advisory group, and the critical intelligence unit. And while they've been very active, these groups, the low incidence has meant that there's been very few opportunities progressed. So Sphere's involvement in therapeutic and vaccine research up to this point on preclinical and early phase studies of various treatments. There's been ACE inhibition approaches, convalescent plasma, hyperimmune globulin, monoclonal antibodies, as well as some support for various vaccines in the preclinical stage, as well as support for the Novavax efforts through the University of New South Wales. We've also placed a fairly heavy focus on treatments outside the pharmaceutical lens, looking at symptom management, including non-invasive ventilation and breathlessness management approaches, clinical safety around things like aerosol generating procedures. We've also invested quite heavily in supporting novel diagnostics and non-invasive and rapid turnaround techniques, as well as a lot of work from our Genome Connect clinical academic group focusing on whole genome sequencing to support contact tracing and microbiology to assess host response to infection and therapeutics. We've also been involved through our partners in a number of the international platform and adaptive trials ongoing, specifically ASCOT, the Australasian COVID-19 trial. The challenge here has been the low case count, only 33 patients across 33 sites since this was established several months ago. We've also had involvement in the pandemic appendix to REMAP-CAP, ICU-focused trial, and have seen a locally established Bayesian trial building on REMAP-CAP in non-ICU settings, looking to assess the efficacy of a number of different treatments outside of ICU for managing pneumonia. While we've not been in a position to contribute significantly to the evidence base regarding therapeutics and vaccines, we've still found much fertile ground for study as a result of the pandemic. And we've really recognised that the criticality of the health system response in that, ensuring that the New South Wales health system is actually at its best capacity to treat. So we began very early with advocacy with the New South Wales government for a focus on health system research to support adaptation of the system and recovery from COVID. We've worked with critical groups like the Agency for Clinical Innovation. We've also conducted a statewide priority setting roundtable exploring issues of relevance to health system research in response to the pandemic, exploring such things as the health system response, patient and community experience, critical medical technologies that need to be explored, the health workforce and policy and governance systems that will be really important in ensuring recovery. We've also done a lot of work supporting cross-centre project development to address these particular questions. So Sphere's involvement in this domain has primarily been led by members of our clinical academic groups with guidance from us within the Sphere executive and our multidisciplinary health service research projects. So on the screen at the moment is an example of the structure that we've followed to support a critical question around evaluation of the whole system response to COVID, and the ongoing management of COVID in the New South Wales community, and the resilience of the system to future shocks. So this is a model adapted from Hannafeld and colleagues and building on the WHO building blocks to unpack how the health system has responded in terms of treatment and management, and work out how best to restructure the system going forward. We've also focused quite heavily on telehealth, looking at models and modalities for specific populations, as well as issues around remote monitoring for patients treated in the community, monitoring things like cardiac function, O2 saturation, and symptom monitoring for community-based patients. We've also begun focusing quite heavily on healthcare worker burnout and ways to foster resilience, as well as public health messaging for culturally linguistically diverse groups, which make up a substantial proportion of the New South Wales population. We've also begun evaluations of non-COVID targeted services to inform how they best recover from the strictures that have been imposed by the pandemic, examples including maternal health and frailty management. And throughout this, we've engaged really heavily with our partner universities, medical research institutes, and service delivery partners to achieve really strong buy-in. And we've done this through providing strategic support to our partnership, enabling and guiding rather than adopting a command and control approach, as many of our colleagues have done. First and foremost, we've really worked to enable what we consider the engine room of our partnership, our clinical academic groups, do their work well. So that's ensuring that they're well supported to meet and communicate, engage effectively with funding agencies, and able to articulate the need to redirect efforts and project funding to address COVID. We've also been ensuring that there's a constant flow of critical information from to our senior leaders via our Governing Council, and ongoing engagement with our research directors within our health service delivery partners. And we've also led collaborative prioritisation and grant review processes across the New South Wales university sector to ensure that they were well focused on critical needs and not wasteful of effort, and supported engagement and implementation with our implementation science platform, which we've seen has been really critical in ensuring that even early phase discovery projects are focused on implementation from the outset, given that time is of the essence in responding to COVID. We've also looked towards future technologies through our Frontiers Technology Code and our Health Hatchery Innovation Platform to identify current clinical needs and fundable innovations and enabling investment in those. So the critical learnings that we've taken away is that the sector in New South Wales has strongly supported a diverse investment of resources and capacity, both in COVID-specific and non-COVID impacts on the New South Wales health system and population. Our local clinical and health services research environment has changed quite dramatically, with a stronger linkage between evidence and policy, more collaboration, and a recognition that implementation is a critical consideration at all stages, particularly with things like medicines distribution, what things need to be considered in terms of, challenges with the storage and distribution of medicines right at the outset so that we can design around them. The integrated response and cross-disciplinary clinical academic groups have been an essential element in terms of meeting the local needs in response to COVID. And we're continuing to engage with the New South Wales government regarding vaccines, treatment and health services research and supporting development of health services research communities of practice here locally. And we're also supporting ongoing COVID-focused clinical and health services research, both financially and in terms of in-kind support within the partnership. So I suppose the key message I'm trying to get across is that we've engaged substantially in work to develop vaccines and treatments, but this has been challenging due to the low incidence and effective health system response we've managed to put up. With our local focus on preclinical and first-in-human studies, little that's been done in Australia will be truly transformative in terms of how COVID is treated and prevented internationally. As a consequence, we've matched or perhaps exceeded our efforts in the treatment and vaccine domain with a robust response to ensure that health system can actually deliver quality healthcare and emerges from the pandemic in the best possible shape. This has been strongly supported by our clinical and research communities in New South Wales, and we feel that this has been an important adaptation in our response to COVID, enabled by our collaborative partnership structure and our primary focus on seeing innovation land in practice as rapidly as possible. Thanks very much for your attention, and best wishes to you all.
Okay, thank you, Dr. Walczak, for your presentation. Thank you also for all the panelists for the presentations. So we've now heard from AAHCI members in Singapore, Indonesia, China, and Australia about efforts that they've been involved in nationally, globally in regards to COVID-19 vaccine, treatment, and response. So now we can enter the Q&A session. Now, as a reminder, please use the Q&A feature at the bottom of your screen. It would be appreciated if you could also indicate to whom your question is addressed to, okay? So let's have a look. Do we have already questions there? Let's see. Okay, not yet. Okay, while waiting, I have a few questions to the panelists to start things off. So this whole COVID pandemic is disruptive to many things. It's changing a lot of things. It's unprecedented. So based on your current experience in trying to do this research and development in these unusual circumstances, do you see any long-term changes to research and development processes in your country that will come out of the COVID-19 pandemic and related research? Perhaps we can start from Singapore.
Thanks very much.
Professor?
I
Yeah.
certainly hope so. I really hope that the entire regulatory processes will change. I mean, and specifically what I mean is that, until recently, the time taken from the point where you have a candidate vaccine to getting into clinical trial is on average 3 to 5 years, and 3 years is actually very optimistic, more likely 5 years, right? Here we've gone, from the point where we make a vaccine to the point where we actually have an interim phase 3 trial in less than a year, right? And it looks like even despite that speed, the safety is looking pretty good, right, in terms of the data. So we should ask the question, why does it take so long to translate a new vaccine or a new drug, right? What are the processes that we have been doing or are required to do, which are actually unnecessary, that do not inform on human safety, in terms of the preclinical studies? What are the things that we are doing now that actually does not inform or do not inform on human safety, and start to take those remove some of those studies from regulatory requirement. And the reason is that is besides just the speed, it also reduce the cost of vaccine and drug development. And ultimately, not only do we get vaccines and drugs more rapidly into the clinical space, but it will also be cheaper.
Okay. Thank you, Professor Ang Oi. Dr. Budiman, how about in Indonesia? What do you think?
Well, I suppose it's pretty similar what Dr. Ng has said, but I would say that in Indonesia it has been very encouraging in terms of research and development, the government attitude, and also universities and research institutions. In that we are all hand in hand in trying to face this condition and try to do as much as we can. So I suppose this is very good in terms of the research and development climate in Indonesia, although I don't like the COVID situation, of course, but for research and development, this is good.
Yeah, there's a silver lining to the COVID pandemic. Okay, Dr. Yingji, how about in China? You're still on mute? Okay.
Yeah. I should say that during this pandemic in China, the government is responds very fast. And as a researcher, we get lots of funding from the university level, city level, and the government level. They do put a lot of effort to try to boost any kind of therapeutics development or vaccine development, and also encourage the collaboration of at least in our university with the other university such as in Israel or Canada or other country to fight the COVID-19 together. And I also realized that the whole government's funding is kind of a little bit switched to more encourage the translational research. As a professor said that COVID-19 it's really bad for the whole world, but for research area and government and the university, and they do put a lot of effort and to try to help with it. So.
Dr. Yodi, you're muted.
Okay. Thank you, Dr. Yingji. Dr. Walczak, how about in Australia?
I think it's been really encouraging to see the way that the entire research and clinical community and the government have come together around a common purpose. We haven't really seen that in response to anything before. Where a need is identified, people have been very, very responsive to it. I think, interestingly enough, the processes that have been established for the rapid approvals seem to have accommodated a little bit more risk in terms of what the government will tolerate. And to mitigate against that, they've put in a rapid recall pathway, which they haven't necessarily had before for many medicines, so that if there is a critical safety issue identified, they can recall and prevent usage within 24 hours of a novel vaccine or a treatment. And I think that's a really heartening advancement, because it means that everybody can be assured that whatever gets approved, safety isn't something that disappears. Efficacy isn't something that disappears from people's minds as soon as it's released out there into the clinical environment. It really encourages post-marketing monitoring in a way that hasn't been done quite so well before. I think also, just as I mentioned, the closer ties between evidence and policy and investment are really heartening to see. That's something that we've not seen from our state or federal governments particularly well in recent years. And so people are listening to one another in a much more effective way and meeting each other's needs in a much more effective way. I really hope it continues. I really hope that it's not something that's just tied to this common purpose that we've all got at the moment. I'd love to see it focused on issues like obesity and AIDS, HIV, and any number of other critical illnesses that affect our entire community after this. It would be really great to think that we move beyond self-interest after this. We'll wait and see.
Okay, thank you, Dr. Walczak. Do we have already a question from the audience? Not yet. Okay, fine. So let me continue. So we've heard from Pfizer and Moderna about their results, and which has been quite encouraging, 94.5, 95%, But we also noticed from Pfizer that their vaccine has to be stored in -70 degrees, which is ultra low temperature freezers, which is not easy to get into the system. So we would like to hear from you in your countries. What do you think would be the biggest challenge once vaccines are available? Dr. Professor Ong, can you start with Singapore? What would be the biggest challenge for Singapore once the vaccine is available?
Yeah, I guess there's several factors, right? I think that would influence how we deploy these vaccines. I mean, I think the results have been extremely encouraging. But I hasten to add that these as observation for over about a 3-4 month period. It is actually very short, right? And we know that even a stimulation of innate immune response, which is not long lasting, could protect you for several weeks after that activation. So I'm not sure how much of the efficacy that we're seeing is because of long-lasting adaptive immunity as opposed to short-lived innate immunity, given that this trial is done so rapidly. But despite that, I think nonetheless the results are very encouraging. So I think one thing moving forward would be, to ask how do we monitor the efficacy of this vaccine upon implementation. So I think Dr. Waljak said this very clearly just now. Right? It's not just about rolling out vaccines. You have to have a way of monitoring efficacy, and if there's any problem, how do you stop vaccines from rolling out? And I think that's something for Singapore to consider as well. I think in terms of the cold chain and all that, because Singapore is such a small country, it's not going to be a problem. But yeah, at the end of the day, though, it's also the price and the adverse events associated with vaccination. I think in the early set of the phase 1 trial data, Moderna's has a little bit more adverse events than Pfizer's, but then it was at that time it was a slightly different construct. So we'll have to await the full set of data to be available to judge which one to invest more in.
Okay, thank you, Professor Ong. Dr. Budiman, what's the challenge in Indonesia once the vaccine is here?
Well, I suppose you know that our country is an archipelago, and it will be a big problem in transportation, not to mention the diverse infrastructure that we have in Indonesia. Some are quite advanced, but in many regions, it's very probably not very much developed. So we have that challenges in our country, and the big population in Indonesia, of course, will become a problem in the availability of vaccine or access to the vaccine by our population in Indonesia. Yeah, I suppose that's the major one.
Being an archipelago. Okay, thank you, Dr. Budiman. Dr. Yingji, what's the challenge for China? Still muted.
Yeah. As I know, in China now, we have also inactivated virus as a vaccine strategy, and I think it's also under a phase 3 trial. So we are kind of very looking forward to see the efficacy of that vaccination. But to myself, since I started to work on mRNA-based therapeutics since 2016, and I think mRNA-based vaccine is a very promising one, and also because of the Moderna and the BioNTech, they already announced and show the efficacy. So I think the challenge for mRNA as a medication, first, it's in the history, there is no mRNA medication in the industrial yet. So this will be the first one. So in China, I think there is no CMC standard for making mRNA based medication. So I think the first important thing is we have to set up the standard. And the second thing, I think it's very obvious, like everybody can notice that the storage temperature and how to keep the quality of mRNA medication stable all the time, it's also a big challenge. So that's my opinion. Thank you.
Thank you, Dr. Yingji. So we heard from Singapore, Indonesia, China. Dr. Wojcik, anything specific in Australian context which will be challenging for vaccine?
I think, particularly with regards to the mRNA vaccines that have very, very challenging storage requirements, we have no local manufacturing capacity for mRNA vaccines. So we have to rely on distribution from countries like Germany and the US. And there's going to be a hell of a lot of international competition for those stocks very early on. And that's going to be a real challenge, I think, even though we've secured in principle agreements with the government to get access to these things. It could be some time before we actually have them on shore. Distribution, I don't think, will be a major problem. I think that our national vaccines policy in relation to COVID has set out a really well mapped out division of responsibilities between our local governments and our service delivery apparatus for the management of distributing vaccines, and also for the post-distribution monitoring. We've got some interesting work ongoing around our patient-held medical records, and also some of the data sets that are being established to monitor uptake and storage and utilization. Sadly, I think, though, one of the challenges we'll face here, and it's part of the Anglo-Western culture at the moment, which is really, really disappointing, is vaccine skepticism. We've seen quite a bit of that emerging, particularly in social media in the past months. I think that there's a lot of concern around how rapidly these vaccines have been developed that's been informed by a lot of scaremongering and not necessarily a lot of science. So I think for us, one of the critical challenges will be the public health communications from all levels of government, from clinicians, from patient organisations, and from the media in particular, about the safety, the importance, and the process for uptake. Because I think if that doesn't get right, you only have one opportunity to convince a populace of the right course of action. So I really hope that our entire clinical community here can come together to make sure that this rollout happens in the right kind of way.
Okay, thank you. So we're almost out of time, and I don't see a Q&A question yet, but so let me just give one final questions to close with, just to get your brief responses. So we're all part of this AAHCI. So the question is more of how can academic health centers in different regions of the world collaborate more efficiently together in regards to development, treatment, and vaccines and response for COVID-19? Any ideas, any insights on that? So let's start again from Singapore. Professor Eng?
I know the answer to that question. It's a great question. I'm not sure I have. I think at the end of the day, the collaboration is key, right, in science, and bring different ideas, different perspective. Like for instance, I'm in the kind of issues that Professor Bella Budiman has described. The distribution of vaccines in Indonesia is not something that I would have thought of immediately, because in Singapore that's not an issue, right? Because it's such a small country. So I think with dialogues like that we now understand, we flesh out different sorts of different perspective and different issues that have to be dealt with, and the more we address these issues, the better the access would be to important drugs and vaccines. And I think at the end of the day, especially even in the ASEAN region, if we can collaborate, if all the academic centers can come together and collaborate, that would be a big boon for vaccine and drug development.
Yeah. Thank you. Dr. Budiman, your insights?
Well, thank you. I think you just wake us up. I mean, to the reality that we have not been, using much of the existence of AAHCI. So maybe we should do something about this. There are so many very good, talented scientists here. So why don't we just try to do something about it?
Okay, thank you, Dr. Budiman. Dr. Yingji?
Yeah, I think I have the same opinion with them, and I think most important is that we can get chance to share the information very on time, and then if there is any resource we can share, that will be great. Yeah.
Okay, thank you. Dr. Walczak, your thoughts?
I'm really closely tied to the idea of multidisciplinary collaboration. I think that that's something that served us exceptionally well at Sphere throughout this pandemic. And I think to the point about, the unseen challenges of distribution as you're developing a vaccine, ensuring that, people from the public health apparatus within a country are involved in elements of the design of the vaccine from the outset, so that you can potentially find a way around those distribution channels in parallel with the development of it, so that as soon as it's approved, you're ready to go. You're not working out the challenges later. Ensuring that, your public health messaging, your nursing staff, all of the people that are involved in ensuring that the innovation lands in the patient's, in the patient's environment as quickly as possible. That's the thing that I really, would love to see happen more. I've seen it happen a lot more throughout the COVID pandemic. And I think it's something that can really be facilitated here between partners across the region.
Thank you. So we can look forward to more collaborations among academic health centers in the regions to accelerate progress for vaccine, treatment, and responses to COVID-19. So on that note, I think we can close this session. Please note for the attendees that the link in the chat box for the last session of the Southeast Asia Regional Meeting later this morning on preparing academic health centers for the future. I would like to thank you once again to all the panelists for sharing your experiences and insights, and to all of the attendees for joining this session. Thank you and enjoy the rest of the day.
Thank you, everyone.
Thank you. Thank you.
Bye-bye.